talk. Tom is a professor at the University of Maryland. He's leading a very comprehensive program ranging from structural analysis of envelope glycoproteins of HCV for the selection of the right immunogens to the delivery of vaccines, nanoparticles, and adjuvants. And so it's the whole package. So we look forward to hearing from Tom.
Okay, thank you, Roger. Let me see if I can get my slides up. There we go. Can you see this?
Yeah.
All right, great. So anyway, thank you for inviting me to give this talk regarding our structure-based vaccine design approach to develop an HCV vaccine. This, of course, has been a major challenge in the field for many years. So in this presentation, I'll give a brief overview of our efforts with a focus on the structure-based guided design of E2 and E1E2 to enhance broadly neutralizing antibody responses, the development of a secreted form of E1E2 that maintains nearly the same antigenic profile as the membrane-bound form, utilization of the most robust adjuvant and delivery system in our hands that elicits neutralizing antibodies and cellular immune responses suitable for clinical use, our efforts to boost the immunogenicity of E1E2 using a multivalent nanoparticle approach, and then briefly discuss our vaccine evaluation and testing plan. If there is time, and I know this is a lot, we can briefly discuss an E1E2 consensus sequence approach versus a cocktail approach of multiple E1E2 genotypes within the same formulation. Now, over the past 15 years, we've learned a lot about human monoclonal antibodies to multiple neutralizing epitopes that recognize determinants on the E1 and E2 envelope proteins. Thanks to the work done by Nansen Law, Stephen Fawn, Justin Bailey, and other collaborators in the field. They have kindly provided their antibodies to us that has allowed us to interrogate E1 and E2 in terms of our structure-based vaccine design approach. Now, the structure-based design work that I'll describe today is based on the E2 structures established by Nansen Law and Joe Marco-Trigiano back in 2013-14, and more recently by Pamela Borkman in 2018. However, we were limited in our approach because we do not have the E1E2 heterodimer structure to help guide our work. We are now in near completion of the structure of the secreted E1E2 by cryo-EM, which is currently at 3.9 angstrom resolution. So this slide shows our current atomic structure of E1E2, work that was primarily done by Adi Ofek, Matt Metcalf, and Eric Koff, along with help from Nansen Law. The CD81 binding interface is shown as white dots on the right, based on the contacting residues identified by Joe Marco-Trigiano's recent E2 CD81 crystal structure. This diagram shows the various cross-neutralizing antibodies that bind to different regions of E1 and E2. These can be linear epitopes as the one downstream of the HBR1 region of E2, domain E in blue, or they can be discontinuous epitopes within E2. For instance, domains B in yellow and D in green, or domain A in red that is non-neutralizing immunodominant epitope. There are other important epitopes that are discontinuous and require the presence of both E1 and E2, E1E2 complex epitopes in purple. These E1E2 complex antibodies are high affinity, they're cross-neutralizing, and bind preferentially to the E1E2 heterodimer. This is one of the reasons why we are keen on developing the E1E2 heterodimerase vaccine. The other reason is that there's clear evidence of protection of an E1E2 vaccine in the chimpanzee model as shown by Michael Houghton and others. So a key epitope that we have focused on is domain D, defined by our collaborator, Stephen Fogg. This region is highly conserved among all HCV isolates, and an antibody called HC8426 is broadly neutralized and then defines this conformational epitope. So our question was whether we can use proline that has an aromatic ring that can lock the backbone of a peptide sequence to lock the domain D epitope in the bound HC8426 conformation. So backbone analysis using Ramachandran plotting and Rosetta modeling identified candidate positions for proline substitutions. Proline insertion at amino acid 445 in the blue box was found to be most favorable in silico, so we proceeded with cloning this in the context of secreted E2. Several other structure-based combinations were also tested that I just don't have time to discuss. So the modified E2 designs were purified as secreted E2 proteins and tested for thermal stability and binding affinity to a panel of MABs, as well as to the CD81 receptor. All designs showed high thermal stability as measured by DSC. To assess the antigenicity, binding measurements were done using MABs that target E2 antigenic domains A, B, C, and D. The binding affinities were largely maintained for wild type and mutant SC2 designs. However, there was a notable 10-fold increase in binding affinity of the SC2 design with a 445P mutation for the domain D antibody H8426. This is highlighted in red. So this is a candidate that we're moving forward with. So the SC2 designs were then tested in vivo to assess induction of cross-neutralizing antibodies. Mice were immunized with these SC2 designs and tested for homologous and heterologous serum neutralization, representing H77 homologous to the vaccine and 6 heterologous isolates. The heterologous isolates boxed in red here represent the most resistant isolates to a panel of neutralizing MABs. Interestingly, there was relatively large intra-group variability in neutralization of homologous H77 boxed in yellow here, and no statistically significant differences among the groups. However, the ID50 values for individual mice vary less within groups for the heterologous isolates, and significantly higher neutralization was observed for 2 resistant isolates, 118.1 and J6, boxed in blue, using the 445P immune sera. This indicated that the 445P mutation helped to stabilize the conservation of E2 to induce a more cross-neutralizing antibody response. So as we know, the E1A2 complex contains key epitopes that elicit neutralizing antibody responses, making it a primary target for vaccine development. However, the E1A2 transmembrane domains that are critical for native assembly make it challenging to produce in a homogeneous soluble form because it needs to be purified from the endoplasmic reticulum of cell extracts. So in order to enable a rational design of the E1A2 vaccine, as well as for our own structural characterization efforts, we designed a soluble secreted form of E1A2, as E1A2 shown here, that is produced at higher levels in the membrane-bound form, MB-E1A2, and is vetted by the cellular quality control mechanisms for secretion, making it perhaps more likely to display neutralizing epitopes similar to the virion. Also, a higher degree of homogeneity may enhance evaluation of the stabilizing mutations relative to the membrane-bound form. So here we show the designs of a set of E1A2 constructs that we tested to determine which ones might be suitable for development of a native-like secreted heterodimer. In specific, this slide shows 3 scaffolds that were tested. In B, a human phosgene leucine zipper, a coiled heterodimer with a known structure derived from its transcriptional regulator in the mammalian cells. And C, a fold-on domain from bacteriophage P4, a self-trimerizing domain used to stabilize other soluble viral glycoprotein trimers such as spike and RCF. And in D, a heterohexameric peptide complex that would reflect a model of E1A2 trimeric architecture. In expression studies, the phosgene leucine zipper was efficiently secreted, so we moved forward with this construct. In this slide, we purified membrane-bound E1A2, secreted E1A2 with a leucine zipper, and a modified form of the leucine zipper with no sequence, human sequence homology that we call SynZip, SZ, that I'll discuss a little bit more later, and secreted E2. We again tested for binding to a panel of MABs corresponding to the major domains, as well as antibodies that recognize the E1A2 complex. Importantly, all binding affinities were approximately the same, except SE2 bound better to the non-neutralizing antibodies to domain A, highlighted in orange, The secreted E182 bound better to the domain E antibodies, highlighted in yellow, and we were able to improve binding of the AR4A to secreted E182 using the same zip scaffold highlighted in green, sort of similar to the membrane bound form. All constructs showed similar binding affinities to CD81. Thus, we were able to demonstrate that we can make a secreted form of E182 that maintained a conformational structure similar to the membrane bound form. So to further demonstrate the identity between membrane bound and secreted E182s, specific mutations were placed in both molecules that would reduce binding to domains B and D in panel A, to E1 and E182 in panel B, and only E1, E2 binding in panel C. These mutations were first identified by Manson Law's group, and then subsequently by our group. Justin Bailey's group also mentions these mutations as hotspots for the binding of the respective antibodies. Suffice it to say that there's a one-to-one correspondence in binding profiles for all these mutants between membrane bound and secreted form of E182, further validating our approach. This slide shows the analytical characterization of the membrane bound and secreted forms of E182. Panels A and B use analytical ultracentrifugation, which can separate a mixture of protein populations more precisely than SEC. Suffice it to say that secreted E182 in panel A is less heterogeneous with 2 peaks than the membrane bound form E182 in panel B with 3 peaks. These peaks correspond to free E2, a dimer petaldimers, a trimer petaldimers, and higher order aggregates. We next use SEC with multi-angle light scattering sigmoids in panel C and D to examine the heterogeneity and size of the secreted and membrane bound forms of E182. The average size of membrane bound E182 in solution is about 1.1 megadalton, and the size distribution is quite broad, from 0.5 to 2.5 megadaltons. By contrast, secreted E182 contains fewer highly populated species in solution in panel C, and is smaller, from 130 to 200 kilodaltons. Therefore, the membrane bound form is larger and probably has more aggregates and a more uniform secreted form. So we next tested for immunogenicity in mice, in which purified membrane bound and secreted E2s and secreted E2 here were formulated in our water-soluble polyphosphine-based system that I'll talk a little bit more about later. We then assessed the breadth of neutralization against all genotypes of HCV using pseudoparticles, as well as infectious virus using the HCV-CC, representing all 7 genotypes. Work that was done in Alex Kloss's lab. As you can see, secreted E182 in the red bars induced broadly neutralizing antibodies against all of the genotypes. On the other hand, membrane-bound E182 in the blue bars and secreted E2 in the green bars showed low or no neutralizing activity against some genotypes such as 1B, 3A, 7A. But overall, membrane-bound E1, E2 was significantly lower than secreted E182 in genotypes 1A, 2A, 2B, 3, 4, and 7. These results were recently published in PNAS. Thus, we can say that immunization with secreted E182 is superior to other antigens in this mouse model system and at the dose given. We next move forward in the guinea pig and a pilot macaque study. In the interest of time, I will only discuss our recent macaque data. Before I proceed, though, I'd like to just pause and tell you a little bit more about our polyphosphine-based adjuvant and delivery system. Ideally, we need an adjuvant system that can prime robust antibody responses, as well as CD4 and CD8 cellular responses using the subunit proteins. This has been a limitation in many adjuvant systems today. In this slide, we show the scope of common vaccine antigens and delivery systems, as well as polyphosphine, or PPZ for short. All systems are particulate in nature. They're salts, they're emulsions, they're seals, they're vesicles, they're insoluble polymers. Polyphosphine forms a water-soluble spoil and has a phosphorus nitrogen backbone that gently wraps around the protein to stabilize the structure. These polymers absorb antigens similar to alum, but are not particulate, and they display antigens to the surface of cells. It serves as both a delivery vehicle to enhance absorption of an antigen to immunocompetent cells, as well as it has immunostimulatory properties of dose-dependent cytokine production. They can be made to have virus-mimicking dimensions, and they can self-assemble with antigens, for instance, E1E2, or E1E2 plus 4, or E1E2 plus peptides, et cetera, and small molecule ligands, unlike other delivery systems. Our lead polyphosphatine antigen, PCPP, has been used in multiple clinical studies. A slight derivative, PCEP, is in preclinical development. PCEP is a more potent adjuvant to elicit antibody responses and gives a more balanced Th1, Th2 response. We can also incorporate a TRL7/8 small molecule agonist called risankimab, or R848, to bolster humoral and cellular responses even further. This is the adjuvant system we use more forward into the MACAC studies. Now I'd like to focus on our pilot MACAC study. So for this study, we used highly purified antigens corresponding to the membrane-bound E1E2, secreted E1E2 with a synzip scaffold, and secreted E1E2 with a synzip scaffold plus the stabilizing 445P mutation that I discussed earlier. The Kamasi stained gel with purified proteins and immunoblobs using anti-E1 and anti-E2 antibodies are shown here. The gels show the high level of purity and the E1 band migrating about 30 kilodaltons and E2 band about 65 kilodaltons. These proteins were then formulated with our PCEP and R848 adjuvant system. For the MACAC study, we primed the macaques about 3 per group with 100 micrograms of E1E2 and boosted 4 times with 50 micrograms over the course of the study. Prior to injection, each formulation was checked by DLS. Interestingly, the secreted formulations gave consistent DLS profiles of approximately 100 nanometers in size, the orange curves. However, the membrane-bound gave disparate profiles of 100 nanometers to larger agglomerates, the gray curves, probably due to the lipid and other aggregate nature of the purified antigen. This may influence immunogenicity since aggregated samples tend to be more immunogenic, albeit more difficult to characterize. This slide shows the endpoint titers over the course of 295 days, in which we see a typical sawtooth pattern of titers after each injection as a function of time. The membrane-bound E1E2 immunized macaques, the blue line, had overall higher endpoint titers than the secreted E1E2 forms, the green and red lines, when tested for binding to membrane-bound E1E2 antigen on the plates. Perhaps this is due to the aggregate nature of the membrane-bound antigen. Also, secreted E1E2 with a 445P mutation, the green line, gave consistently higher titers than the secreted form alone, the red line. And we have seen this before in previous mouse and guinea pig studies. Interestingly, the peak titers in all groups seem to level off after the second boost, as shown here. This slide shows the overall endpoint titers at day 105 for the 3 groups when both membrane-bound and secreted E1E2s are used as capture antigens on the plates. Membrane-bound E1E2 immunized macaques responded more favorably to its own antigen when coated on the plate, black bars, versus when secreted E1E2 is coated on the plate, red bars. Similar endpoint titers are observed from macaques immunized with membrane-bound E1E2 and secreted E1E2 with a 445P mutation when secreted E1E2 is coated on the plates, comparing these red bars here. Therefore, the serological response to the immunizing antigen is highest to its own antigen when coated on the plates. We are currently evaluating the breadth of neutralization in this pilot study. Here we show individual macaque neutralizing antibody responses depicted as percent neutralization using a 1:40 dilution of serum. We see cross-neutralizing antibody titers across genotypes 1A, 1B, 2A, and 2B, the highest being to genotype H77 that is homologous to the vaccine. It was difficult to obtain ID50 values for most of the heterologous isolates due to the overall low neutralizing antibody levels. Nevertheless, we do see cross-neutralization in which membrane-bound E1E2 appears to perform the best, at least under this particular dosing regimen. And secreted E1E2 with a 445P mutation appears to perform better than secreted E1E2 alone. We need to repeat the study, optimizing the formulation conditions and to increase the valency and size of secreted E1E2 so that the physical parameters are more similar to the apparent aggregate nature of the membrane-bound form. So in order to improve the size and valency of secreted E1E2 and anticipated immunogenic potency of the secreted E1E2, we are making nanoparticles using the spy catcher system for the irreversible conjugation of recombinant proteins. The peptide, 13 amino acids, spontaneously reacts with catcher protein, which is 12 kilodaltons, to form an intermolecular covalent bond between the pair. We are evaluating the ferritin, MI3, and I50 systems at the present time. This slide shows some of our initial results using the MI3 spy-catcher nanoparticle scaffold. In panel A, the coupling efficiency is greater than 75% in all three reactions using a 1-to-1, 1-to-3, and 1-to-5 nanoparticle to secreted E1E2 ratio. In panel C, our initial negative stains are shown here, where we can show clear decoration of the nanoparticle with secreted E1E2 as shown in the red circles. In this polyvalent state, we would expect the avidity effect leading to tighter antibody binding for the nanoparticles relative to secreted E1E2 alone. In panel D, we observed improved KD values for all four MABs tested, but most notably for AR5A to E1E2 and 526 to E1. Finally, our vaccine testing and evaluation plan is shown here. We plan to test our top vaccine candidates shown in the red box on the left, which depicts a secreted and modified E1E2 as a nanoparticle, and its membrane-bound E1E2 counterpart as the top vaccine candidates. In order to demonstrate protective efficacy, in collaboration with Alex Kloss, we will use a human liver mouse chimeric model, immuno-incompetent, and humanized mouse model, immune competent, to demonstrate protection against challenge with infectious virus. The chimeric model will receive passive transfer of vaccinated metaxia prior to challenge. The humanized mouse model will receive active immunization with the vaccine prior to challenge. We will test for protection in a tiered fashion, first testing with homologous virus challenge, and then testing with more genetically diverse strains. Our lead candidate, once it is identified, our plan is to demonstrate safety and efficacy in clinical studies using high-risk patient population of people who inject drugs and show prevention of infection or reinfection, and then the vaccine could then be rolled out to other high-risk groups. So, and finally, I'd just like to thank all of our collaborators and funding agencies that were really the drivers behind this research. It has been a team effort. Thank you.